Hi there, welcome to Live from One Cancer Place. Today we're really pleased to welcome Dr. Scott Kopetz from MD Anderson. He is one of our very favorites in our cancer patient land world. He's kind, considerate, generous, and humble. And you will learn that in the conversation that he is going to have with Dr. Chris Heery, who is our host. The two physicians have a grand time back and forthing about the current state of the art for cancer treatment and cancer research. I am Erica Hansen-Brown. I am the founder and CEO of One Cancer Place, which is being styled as the gateway to patient land for the brand new cancer patient. Basically, what we want to do is be the guides for the new patients and help them to find friends, companions on for their journey, and also lots of information that's relevant to their treatment, their successful treatment. So take it away, Dr. Chris, and introduce our wonderful guest, Dr. Kopetz.
Thanks for having me again, Erica, to do this. We're really lucky today to have Dr. Kopetz to do this conversation with live with One Cancer Place. So Scott, let's have you just sort of give an intro to yourself and what your main areas of work are. And the first question to lead into that you can roll into sort of your intro is, what are some areas that you think are really critical right now in cancer research, particularly in the tumors you're working on most?
I'll start off just by saying I'm a very simple man, and I have one tumor that I work on, and that's kind of colorectal. So that makes it easy. But then we've learned that things are very complex, and that's not just one tumor type within colorectal. So that we can certainly spend some time talking about that. So my name's Scott Kopetz. I'm a faculty professor of GI Medical Oncology here at MD Anderson Cancer Center. I've been at MD Anderson for about 17 years. Did my training and actually my PhD in cancer biology here, but did earlier training, my MD and internal medicine training at Johns Hopkins and Duke before coming here to Houston. I'm one of the leaders of the GI program, the deputy chair for the department here, and have really been focused on trying to really improve research in colorectal cancer. I'm a physician scientist, meaning I see patients as well as have a lab that focuses on developing novel therapies and kind of understanding the biology of colorectal cancer. It's a lab that is very translational in nature, I mean, very close to the clinic. We're trying to learn as much as we can from the patients and the patient material that we obtain from clinical trials, models that we develop out of patient tumors as well, and some of the both mouse models and more kind of traditional modeling that's done. So it's other roles I think that are involved in the NCI's trial network, and I chair the colon cancer task force of the GI steering committee, which helps make and identify priorities for clinical trial research within the NCI trial system. I also lead the moonshot for MD Anderson in this space. So really thinking about within the realm of colorectal cancer, what are the areas that we really should be prioritizing and opportunities to really kind of look at kind of innovative spaces and ideas, and happy to kind of share some of the efforts ongoing in that space as there's interest or time. Yeah. So a great question, Chris. Like, what's exciting, right? What are the areas of excitement in the field? You know, and it is an area where we're seeing progress. It's not as much progress as we'd like. Never is. We have recognize that we have some therapies coming in, but what we've kind of seen over the past few years are kind of nibbling away at the rare, subtypes of colorectal cancer. And I think, what we're missing are really kind of opportunities to more meaningfully advance the majority of colorectal cancers and improve outcomes there for patients. So it's, in my mind, I think there's a few key areas. If I had to kind of highlight a few of them, I'd say, one of our key areas for, and we're excited about, is thinking about how we can utilize novel biomarkers to really attack cancers earlier when they're much smaller. And that's kind of in the setting of what we call minimal residual disease, ctDNA. So that would be kind of one area. The other is that really the evolution of biomarkers. And I think as the title of our session, it's really about how do we get beyond the defining colorectal cancer on the basis of their mutations and really start to think about how do we start to define colorectal cancer on the basis of their behaviors and at a genomic level. And so we talk about the transcriptomics or the RNA behaviors of the tumors, and how do we utilize new ways to classify colorectal cancer that may help us really decipher the subtypes of colorectal cancer that are meaningful for therapeutics. And then I think fundamentally this is more of an abstract concept. Is I think we have a reasonable understanding now of first layer or two of colorectal cancer biology when we're looking at primary tumors that are untreated. And I really think that there's a need to go deeper in the field to understand how our standard of care treatments change the tumor. What is the biology of a treatment refractory colorectal cancer, right? So there's this gap in the field between, what we think we know about the cancer, and then when we try a new therapy in a patient who may have progressed on 2 or 3 lines of therapy, and the cancer is not behaving like we think, well, it's a bad drug. Well, or it's that we just don't understand how the tumor has evolved as much as we'd like, right? And so really trying to understand how a metastatic tumor that's seen treatment, how that differs. And I think there's a lot that we need to understand there, but I think some efforts being made. So those are kind of 3 very broad areas, Chris, and I'm sure we could think about a few more, but it's really, ctDNA driven disease, RNA-based classifications, and then, understanding the evolution of colorectal cancer.
Yeah, and I'll just maybe comment here for the audience that those 3 things are actually fairly tightly bound together through some core principles, right? So, you might be watching this and saying, is this all going to be about colon cancer? And the answer is, we're going to talk about colon cancer for the most part, because that's what Dr. Kopetz does. But the concepts and the principles are applicable across essentially all cancers. So first, looking at gene expression, Dr. Kopetz was on a paper that I think is really one of these core papers that should change the way we think about how we manage cancer, particularly colorectal cancer, but I think it will end up applying to a lot of other cancers. And what that paper looked at was this RNA expression profile. So we've done this on a previous one of these conversations, but just to remind everybody, the link between DNA and RNA and protein is that the DNA is the code, the RNA is sort of how that message gets conveyed, and then that gets turned into the proteins that actually make cells function certain ways. And the proteins are really what determine how cells behave. So anyway, I'd love for you to kind of opine a little bit on how you think about biomarkers and tumor selection or patient selection, and that evolutionary process, and how you can understand that process as it goes from, first treatment to second to third and so on. How are those concepts are related in your mind?
Yeah, absolutely. I mean, I think that that one of the findings that we see, and I think, as Chris, as you nicely outlined, when we're thinking about the DNA, the mutation status of a tumor, we recognize that can help us, think about what general neighborhood this tumor lives in, right? It behaves, like a tumor that, lives on the Lower East Side, right, or of the city, but it doesn't really tell us anything more about that behavior, and that you have to kind of think about how the tumor as the tumor evolves and grows, that those DNA mutations don't really change a whole lot. There are some examples where you can acquire resistance to certain very specific targeted therapy, but in general, we think that these mutation patterns are really fairly stable over the life of the tumor, and the evolution of the tumor across the different stages and settings. And so, but at the same time, as clinicians, we look at it and we say, well, this tumor responded, when we gave this treatment first, and then it stopped responding, and then it moved to a different area of the body and behaved in a different way, and then maybe didn't respond to this therapy when it was given again, and when we profile it, we say, but the DNA is still the same, right? The fundamental mutations are still unchanged between all these different sites. And so I think this is, as Chris mentioned, is recognizing that it's really not that the instruction manual has changed, it's the interpretation of the instruction manual, right? It's how the tumor is adapting and evolving to this, and it may be interpreting the instruction manuals in a very different way. And as a result, the RNA is very different, making very different proteins and having very different behaviors, right? And so I think we have learned a lot, and I don't want to discount the tremendous advances that have come out of profiling the DNA. And this absolutely should be a part of so many of our solid tumors, including colorectal, where we say, everyone should be getting could be getting these biomarkers test, this DNA tested. But that we recognize that doesn't fully define the disease. And I think that's where, as Chris, as you mentioned, it's really about recognizing that the behavior of the cancer is really tied to, other metrics beyond this mutation. And so I think that's where we're starting to learn more about how the RNA and the protein levels adapt over time. But it's still really early, right? And but we're starting to get to the point where there are tools now available. Like, as a patient, you can go and you can order an RNA profiling of your tumor. Right? It's not common, but it can be done. And then the question is, well, what do we do with that information, right? And this is a much more complex set of information to decipher, to interpret, and kind of turn it into something that's kind of clinically relevant.
I am also interested, though, in how we think about those things in that evolutionary process, because one of the things that I think is really a relatively difficult concept until people have thought about it a bit, from the patient perspective, is this idea of evolution of a tumor. So can you talk a little bit about, just how you think about tumor behavior and how it's shaped over time by, what pushes it one way or another?
Yeah. So there's two different types of tumor behavior and evolution that we kind of think about very broadly, right? So if we think about an untreated tumor, maybe it's a patient that has disease that had spread prior to diagnosis, so a stage 4 metastatic patient, newly diagnosed. We have a number of different tumors, cells in there that are heterogeneous, right? Now, what we think for most colorectal cancers is they undergo what's called a Big Bang evolution early on, right? This is not true of all cancers, but for colorectal, we think this model applies. That means that there's some point that you get very early on before diagnosis that you get multiple, a few alterations, key alterations, mutations, a cancer forms. And then there is a big explosion of new mutations that appear in the tumor that appear and then are present in a lot of, the vast majority of the cancer cells. The tumor then grows. It doesn't acquire that many more mutations. It can pick up a few heterogeneous mutations, but those mutations are present. The tumor then at some point spreads out of the primary, carrying those same mutations to these different parts of the body. So the mutation patterns seem fairly different, but each of these different cells have different behaviors nevertheless, right? They have different different states, as we call, that they can be in. And these different cells comprise a fairly heterogeneous population. And we know that because we can take these cells, all of them have, the almost identical mutation profile, right? We can treat them with chemotherapy and maybe we get a great response and we kill off 90% of the cancer cells, but 10% of them survive. Now, why do they survive? They have the same genetic mutations as all the others, but they have a different transcriptomic state. They have a different RNA state and a different behavior. So one type of evolution that we see is this kind of pruning effect, right? We treat the tumor, some parts of the tumor die off because those are the sensitive cells, right? And what's left then becomes the cells that then can start to grow and divide, and those are the ones that are resistant to the treatment. So that's kind of one type of tumor evolution that we see. But the other is this kind of adaptation of the tumor cells, right? So that when you treat some of these cells, some of them can undergo state changes. It may not have been that they were innately resistant to that treatment, but they can get stressed and they can turn on programs, survival programs in the tumor. There's a number of these, diapause, epithelial mesenchymal transition, all these kind of terminologies that we throw around in the literature to describe how the different tumors, the different states that they can undergo. And that these different states, then this kind of evolution of individual clones is not pruning so much, it's just these individual clones are then changing. So you can see how you're then left at a time of progression for a patient where you have some clones that, some subtypes, some subset of the tumor that had been killed off, some subset of the tumor that has evolved into resistance, and some subset that may have been resistant from the very beginning, right? And so that's the complexity of this when we think about the evolution of the tumor, all of while really keeping the same mutational profile, right? And that's the level of detail that we need to get down to as a field, right? Go beyond the DNA to start to look at all of these changes that are occurring.
Yeah, I mean, I think the point that I'm trying to bring home for everyone is the complexity of how many things need to be measured to understand this a bit too. Because I want to come back to the minimal residual disease and circulating tumor DNA concept. But I think one of the things that's really important for the audience to understand is that measuring, for instance, the genetic mutations, so the changes in the DNA, is something that's now capable, now that can be done at a relatively low cost and can be done relatively similarly across many, many labs. And it can be done within, a couple of weeks usually. But I will point out that oftentimes when we think of this, we are imagining that we're looking at every single base in the DNA, every single line of that code, every single 0 and 1 in the code. And we're actually usually not. We're usually looking at things that are already within panels that we believe are relevant in that given disease, or they can be a very, very large panel, but they're not absolutely everything. So they can miss things. And that actually comes to this other point that Scott's been referring to, which is the expression of genes can actually be changed by selection pressure, things that force a cell to have to do certain things to live, meaning that it'll receive a signal of something in its environment, and it'll accidentally upregulate some other gene, and it'll accidentally survive, and then therefore will have a survival advantage. Or there can be things in the parts of the genome that we don't measure that could be changing the expression of certain genes that we do measure, right? So that's why we have to measure the next thing downstream, which is that RNA part. And so, I mean, you can think about this almost like, you can see two sets of identical twins, and you can see how they look sometimes a little bit different from each other, right? They just, maybe they ate slightly different diets, or they were in the sun a little bit more or less, or whatever it was. And you can see that they're slightly different in their appearance. And that's not different than what we deal with in cancer cells. The difference is that we're dealing with hundreds of millions of cells or billions of cells, even at the earliest stage of that disease when we're trying to get rid of it, right? So we have to account for all that heterogeneity. And that's what I wanted to bring that point back to, this idea of circulating tumor DNA a bit, and the concept of, how do we know that we're capturing the right signal when we're measuring circulating free DNA or any other minimal residual disease, or are there limitations there that we have to accept and just realize that we're trying to get the most benefit for the most patients? Could you talk a little bit about that concept? So now we're definitely in the deep weeds.
That's right. Yeah. No, and it's, and to wrap up a little bit about the RNA before doing that, I would say, so the question naturally is, okay, great. You've convinced me, Chris and Scott, that we've got to dive into the RNA, right? Why can't we do this and make rapid progress? Look at all the great progress we made at DNA. DNA tends to be a little bit of binary, right? It's present or it's absent, right? Mutation is present or it's absent, and and like I said, they're, with some rare exceptions, they're pretty static over a long period of time with the tumor. RNA is not just dictated by a handful of, maybe, we think about DNA mutations. Maybe there's 100 critical DNA mutations that we measure and know what to do with right now in the clinic. RNA, really, you've got to take a look at the total of 20,000+ genes, and they're all continuous levels, and they're dynamic, right? So it's a huge number of genes, very dynamic, right? Where they're changing. It's not a 1 or a 0, not binary, but very different levels, and they are changing frequently over the period of time. So that's where it becomes difficult, and where this is not for the faint of heart, but this is where really kind of some of the deeper interrogation and these efforts have to go. Now, so I think there will be advances in this field, but I think, it's going to take some time to really understand and decipher all of that. And they're going to be, require larger amounts of effort. Now, the one way to assess some of these changes is instead of sampling the tumor directly, is looking at circulating tumor DNA, right? The idea you can profile tissue or profile plasma, looking for the detritus of dead and dying and stressed cells from the cancer, and you can find these things. Now, you can find DNA, right, that's present. And you can also find RNA there as well. RNA, for all the reasons I mentioned, is trickier to interpret, has a little shorter half-life in circulation, it's much noisier. So it's there. We don't know what to do with it yet. It's still a little complex to sort out. For the aficionados out there, there are some, there are little fat bubbles, exosomes that hold some of these materials from the cancer cells, and you can profile the RNA out of them. But the DNA tends to be hang out a little bit longer, still not very long in circulation, like a few hours, right, after it's released. And we at least know a little bit more what to do with, because we can kind of look at a few of those. So the questions are really about then how do you utilize that test to really ask some questions. And there's a number of questions that you can ask with circulating tumor DNA, right? You can ask, how did the tumor evolve over time at the DNA level, right? So before I mentioned, yes, you can see tumor change. It can acquire a KRAS mutation, right? Acquire an ectodomain EGFR mutation in colorectal. EGFR mutations that can be, acquired and changed in lung cancer. So a number of these different things, can occur. That tends not to be the big driver, except for these, few examples. Doesn't really represent the totality of the changes over time. Most of the time in colorectal cancer, I'll check a liquid biopsy, a ctDNA later in treatment to try to understand how the genomic profiles changed. It's the same, right? The DNA level really has not changed at the genes that we're profiling, to Chris's point. But although we know the behavior very much has. So, how do you monitor treatment over time? Can you use the level of ctDNA to define whether or not a patient's responding to treatment? Give a treatment, does that level drop? Just like you would a protein marker like a CEA or a CA125, something along that line in ovarian cancer. Or, and this is what I was alluding to at the very beginning, can you use that to determine whether or not somebody still has microscopic disease somewhere in the body, despite, a surgeon completing it, we got it all, right? The adjuvant therapy that may try to clear all that out. Can you then use that to, and despite a CT scan that says we don't see anything in the body, right? But yet we know, given enough time, cancer recurs, meaning it was never really gone, right? It was there all along. It was just below our limits of detection. So that's where we see a lot of promise of this circulating tumor DNA to really assess, is there disease that remains behind that we can't detect by our current imaging data and imaging tests. Now, as Chris mentioned, there's questions about, how do you do this, and how, in the setting of tumor evolution and heterogeneity, do you make sure that your test is behaving correctly? And that's an area the field's working on currently.
I think we've touched on some really, critical topics, and I want to maybe take this direction towards sort of how do we apply this, and how do you get into this field? How do you pull patients into these conversations? Could you kind of walk us through, maybe you can think of an example from, the last month or two of clinic where you've had to try to tackle some of these issues and some of the struggles patients have had to understand it, or, how have you brought that around to a level of understanding that could actually help patients?
Yeah. So how do you think about, how does one think about all of that? I think the key is that having these conversations about what they mean and what they're for, and what we're going to do with that information. And so I'll share one example of kind of probably the most common discussions that we're having right now with circulating tumor DNA. Right, so a patient that may have stage 3 colon cancer. Has received, good surgery, received 6 months of adjuvant chemotherapy, perhaps, and we're kind of in that surveillance period. CT scans are done. They don't show any evidence that there's disease that remains. We're obviously very hopeful that there's no cancer that remains, but we know that, a proportion of patients, maybe 30% of patients, will have disease that remains, meaning that it will recur at some point in time. Now, the one question is, well, could we do anything else, right? Common question. Well, what if we gave more chemo and, or did something else? And here we just say we don't, right now, the evidence is, unless we know what we're treating, there really is no role for any additional therapy. So then we start to have this conversation with the patients about, okay, we have a test that can potentially tell us whether or not there is microscopic disease somewhere in the body, right? The test is not a perfect test, right? It can miss it. There could still be disease there and it could miss it, but it's pretty good that if we see it on the test, we can be fairly confident, 95+% confident that it's really there, right? We don't exactly, there's no standard of care that we would do on the basis of that result. And then we start to talk through, well, is this going to be helpful, right? And this is a conversation that has so many different levels, right? It's kind of what does the patient want to do? What are the options that we have in this setting to actually act on it? And as I mentioned, colorectal cancer, there's really nothing that the standard of care, the guidelines would say do. You just watch, and if something appears, if it's resectable, great, you can resect it. If not, you have to wait and initiate systemic therapy for metastatic disease. So we have a big effort at MD Anderson that we've built over the past year or two to really have a number of therapies to tell patients that, if you are ctDNA positive and you have no evidence of disease in the body, there are a number of immunotherapy studies that we have. There's some, vaccine studies. There's some lifestyle intervention. There's some cellular therapy studies, some immunotherapy studies that are coming. So there's some options that we can engage on. And so it's really a conversation with the patient about, we don't know that this is going to, that knowing ahead of time that you have disease that remains is going to be helpful. But if you're the type that patient that kind of wants to know, right, recognize that it may cause anxiety, it will cause anxiety, and certainly if you're positive, right, because, we know there's disease still remains, but would be interested in kind of trying to think about, can we do something a little out of the box? Can we try to tackle the disease before it shows up and becomes metastatic? So there's a big hope that these tumors are very different when they're micrometastatic, right? And there's good data to suggest that a lot of the immune defenses that the tumor throws up really is dependent on there being, a certain bulk of tumor, right, that's present. And we detect cancers typically when there are about several million cancer cells, right, by CT scans. So what if we can detect it when it's 10,000 cancer cells or 100,000 cancer cells, right? Is there an opportunity to intervene with therapies that may not work as well when it's really bulky in a billion cancer cells, but maybe there's an earlier time to intervene. So those are the type of conversations that we have. And as you can imagine, there's a number of patients that say, yes, sign me up. I want to be aggressive. I want to tackle. I want to do this. I understand. And there's others that say, you know what? I'm going to come. I'll do my scan every 6 months or a year. But it is going to be what it is, and we'll tackle whatever that comes. And that's completely okay as well. And so this is really a area where we really are encouraging a lot of this kind of conversation, because this is like any new technology storming into the scene. A lot of people are getting tested without really thinking through what it means to be tested and what the implications would be if positive or negative. And we don't fully know what to do with the results yet outside of these clinical trials.
Yeah, I think that's that last comment is really what leads us to, actually one of the purposes of these conversations is, in your opinion, how much effort should a given patient push into trying to have these more complicated tests performed outside of clinical trials? Is there any value there in your opinion? How do we, if there is a value there, how do we make sure that those things get aggregated the right way? Do you have any ideas about how patients can participate in the process a bit more?
Yep. No, absolutely. So I think, first level is just understand the testing that's been done, right? How is that being utilized? How is that being incorporated? And make sure that everything that should be done is done, right? And every time we talk about how to advance the field of biomarker testing, we have to acknowledge that there's a large minority of cancer patients that aren't always getting the biomarker testing that even the standard of care guidelines would recommend. So got to advocate for yourself, be aware of the type of biomarker testing needs to be done, and make sure you're doing that. Then the second question becomes, okay, what about additional tests? Like I have read on the internet about X, Y, and Z, or, the doctors suggested this additional novel therapy. And I think there's a few questions to ask as a patient, right? One is, how will this result be used to change my therapy, right? Pretty standard question, but you'd be surprised how many tests that are out there right now that the answer is it won't, or we don't know, right? And so, that's fair to say for all the RNA-based testing out there right now, for example, a proportion of the ctDNA testing that's out there. How will it help me? We don't know, but here's how you have the conversations around that. And I think acknowledging that that biomarkers can have a substantial amount of benefit in helping to guide your therapy, but as the complexity of the biomarkers rise, these diminishing returns start to occur, right? Sometimes patients will come and they'll say, well, we had a test done that looked at 50 genes over here, and this other test over here will run 400 genes. Is that 8 times better? And the answer is no, right? It's maybe 2% better, right? It's not 8-fold better there, and it's because of those 50 genes hold the vast majority of the information that we know what to do and to act on, right? And as these tests start to get more complex, we start to add more genes, the information that comes at the patient and the care team is much greater, but there's really diminishing return on that. I know the concept of I'm just going to sequence the totality of my tumor sounds like a good idea, but the amount of information that's generated versus the actual return on being able to utilize that is really not there. And this is a little bit of a soapbox here, so apologies as I rant just for 15 seconds or so. But it's a shame how the biomarker field has been pushing better, bigger tests at the expense of really improving the annotation quality of what comes back, right? If they put just as much effort into helping interpretation of the results of their existing panel, both for patients and for physicians, they would be so much more utility out of it as opposed to putting all their energy into making bigger and better and larger tests, right? So that's been the big barrier, is that and part of it is there's no, you can get paid for doing a bigger test. You can't get paid for doing a reimbursement. You can't get reimbursed for doing an interpretation, right? There's just not a great mechanism for doing this. There's a lot of kind of, perverse economic incentives that don't drive the right behaviors in biomarker testing. But I think that that's an important thing to kind of have that conversation with your oncologist. How will this next test help us together as a care team, patient and physician, make the decisions to move forward?
Yeah, I think therein lies the struggle, right? I mean, so there's a need for patients to be participants in this process and to advocate for being a part of getting the sort of next generation of testing. But there's also, I think, without putting any moral or ethical opinions on anything, there are definitely versions of tests out there that are less helpful and more expensive, right? So how do patients sort between those things, and how do they do that without personally seeing you? Because if somebody's at their local oncologist and their local oncologist is not a world-renowned expert on these topics, how do they find the right information to know, should I use this test that claims to have all this stuff, is that really helpful or not? And, what do you have any recommendations for that? Or should people tune into One Cancer Place and listen to these, and then we'll put up the right resources on a website at some point? I mean, is that the only solution at this point?
Yeah, it is. It is tough. And I think that, part of it is kind of understanding what a minimum biomarker testing should be for your tumor type, and making sure that that's done, right? I think encourage absolutely everyone to do that. And I think there are resources out there now for patients to go and define that for various tumor types within advocacy groups to find this. Not always easy to find, but it's worth the effort, right? So I'd say absolutely should do that. And then, as you say, Chris, it's a matter of, okay, well, how do you start to think through these more kind of complex next generation tests? And I think, I think having the conversation before the testing's done is probably the most important thing. So it's not about, hey, can you order this from patient to the doctor, or the doctor saying, oh, by the way, I ordered this, right, on you, right? But it's like, okay, let's talk about what sort of testing can be done. And that's a, worthwhile question to sprinkle into your oncologist visits. Is there any other biomarker testing that would be of, help in my cancer at this point? Right. Kind of put that in your repertoire to ask every so often. Now, it turns out that, it's not something that, that's testing every few months, isn't always as helpful, but it is worth kind of revisiting that question every so often, just kind of given how the field changes, and how that's done. And then I think just kind of being aware that more is not always better. And I think just understanding that concept, that it's the interpretation and the usability of the data is so much more important than the sheer amount of the data, I think is critical. I will say that there are a lot of kind of direct to consumer or patient, testing out there that's not, I wouldn't say, the gray zone, but there's a lot of like, we're going to, do some metabolomic profiling of your cells, or, we're going to, do more in-depth, culturing and prediction of things that will work for you, that invariably, in my experience, have ended up with, worse outcomes for patients, right? So just be aware that there are scenarios. So, for example, somebody who sends there's some cells off to have some testing, and it, comes back that says we need to give, mitoxantrone, or, some, therapy that you'd never use in that tumor type. And we see reports like this, and patients get treated, and they don't benefit, they derive toxicity. So, some of these predictive assays out there in terms of, kind of tumor prediction, treatment prediction ones are ones you don't really see in the guidelines. Most of the time aren't being done. And there is real harm in that, because if you do those and then you're acting on them, that's where I've seen the most harm come out of biomarker testing. Right. So just be aware that it's not always a benign, well, it can't hurt to get the information, because it can.
Yeah, I think that's a great point. I mean, I think we're definitely at the point where too much information can also be detrimental. So I think what we're trying to encourage here is a community of patients who can be a guidepost for others. And so that does mean that those who are willing to take a leadership role as a patient in this and help explain to others, you're also going to have to help sort through some noise. And that means when you meet with somebody who is an expert in these things, that your voice should be weighed heavily, but you should take very careful notes and try not to overcommit to too much information. One thing we do know about a lot of this is we still know less than we will know in the future, and we have enough tools to confuse ourselves. That is for sure. So it doesn't mean that this is a pathway, an effort that shouldn't be undertaken, because the compilation of all of this information is going to be extremely useful over time. And one of the things that we really wanted to talk about today, and we've touched on a bit, is being able to determine which patient is in which category, so that we can design treatment plans for that patient that make more sense. And one of the ways that happens is that if you can identify markers that, sort patients into smaller groups from the huge group, then you can evaluate each of those subgroups in clinical trials specifically for that group. And that might mean you actually use different drugs. I think what we're not advocating for is using different drugs for individuals without ever testing them in a clinical trial, because that now you're getting into a little bit of hocus pocus. And sometimes that works, but more times than often it does not. And it can be very harmful, actually. So what we're hoping is that by people committing to this and participating actively, we will gather the right information. We'll be able to aggregate patients into groups that make more sense, into clinical trials that make more sense. And then over time, all of the outcomes will improve for everyone. But it will take time, and it will take quite a bit of effort on patients' parts to be part of these things. So, Scott, any other topics that we haven't really hit on because maybe we could take a couple questions. I know you're pretty comfortable taking questions from patients. So
absolutely. Yeah.
Patient advocates, are there any? I see one in the chat. So Janet asked, are the direct to consumer tests that are being marketed, how do patients get recruited to do that? Are patients able to actually contract directly with a company, or do they almost always have to go through a doctor to get those things?
Yeah, you can see some that especially outside the U.S. or like where there's U.S. to X outside of U.S. where that can be done. The rules are very different in different situations. I know in Germany there's opportunities for more testing, for more tests like this without a physician being involved, is my understanding. So there are some differences. I think most of the time it's a patient that that's kind of seeking out, understandably, seeking out what more can be done. And there's absolutely nothing wrong with that. But I think, a lot of times it's the, doing the internet searches to kind of look for this, or it's, well-meaning friends or relatives or otherwise that can kind of provide that information. And I think, definitely wouldn't want to discourage getting information and learning, but I think just having a little bit of a caution to recognize, and I think the message of, some biomarker testing, if it's not really well thought out or well done, can be harmful, right? Just recognize that, right? That you want to, just like you wouldn't buy something off of the web and start taking it on a regular basis because, it sounds like a good idea, right? You don't want to go and run a biomarker test off the web if you haven't really done the research and talk things over with your doctor. So I think if we just kind of think about it in that way, that helps mitigate some of that. But I think most of the time it's not necessarily that there's other means. It's typically patients trying to get information and coming across these.
Yeah, I mean, you just said something that made me think, which is the idea that we wouldn't want to just order something off the web and take it without a lot of evidence. I mean, it sounds a lot like the last 2 years, to be honest with you. So I guess how do we deal with, or what advice do you give patients when they're getting just unlimited inputs from family members who all think they read some miracle cure? And how do you handle those situations?
Yeah, I think it's really kind of giving the patients permission, to to not feel like they have to engage or respond to all the advice, right? And that's hard because you have family and loved ones who really want to help, and they're going to do all they can and throw things your way to try to be helpful. And I think the key is just to, feel very comfortable with your treating team, that you can have these conversations, right? Which means as physicians, we need to be open to having these conversations and not just dismiss them out of hand, but kind of, hear them and discuss them and be open. So I always try to do that with patients, like, tell me what you're taking, right? If I'm doing us both a disservice if you're not comfortable enough to share what you're doing outside of traditional mainstream medicine with me, right? I want to be a kind of a partner in that and walk through that with you. So I think, making sure that you have that type of that discussion with your oncologist about that type of information that comes, and really just give yourself permission to, politely dismiss or ignore advice that comes in. Right. I think that's the other key thing.
Yeah. I kind of asked you what I think is one of the harder questions that's out there, though. I mean, this is that there are patients who don't feel like they get enough support. And then there are patients who feel like the support they're getting can be sort of negative influence. And it is a challenge. And I would just support that the one thing you should remember is it's your life, and you're the one that gets to make the decisions about it. And if somebody thinks that they know better than you do, ask them if they would trade places with you. And they're probably not going to be willing. So it's your call. And you really, obviously you want to listen to your family when it's welcome and when it's useful, but you also have the right to tell them to back off and let you make your own decisions. So, and if whoever's watching, if your family's anything like mine, I can tell you that would be very difficult, but I would encourage anyone with a family like mine who's very in each other's business to tell everyone, look, I'm going to be making these decisions. Just give me a break. All right. So here's a question that came from Julie. Great question. So even with a small set of actionable mutations, about 5 for specific cancer types, and in this case, we could be talking about colorectal, we could be talking about lung. There are many nuances, and it really multiplies across cancer types then. All those nuances and how those mutations impact, even if in some cases where the mutations are the same, you can have different treatment or different outcomes with certain treatments. So it seems dissemination of the latest information across the oncology community is still pretty difficult and inefficient. What's the best way for patients to get confidence in their oncologist's knowledge and collaboration with other specialists? What kind of questions do you recommend people ask to try to figure out whether they can trust their oncologist?
Yeah. I mean, and just to reiterate the fundamental part that I mentioned before, because I just think this has to be one of the key messages that we as an advocacy community really push, is just ask, is biomarker testing appropriate for my tumor type, and has it been done? Right. Those questions, I think, can really raise the level of kind of knowledge and engagement. I think, as Julie mentioned, it is really tough, and I'm so fortunate that I live and sleep and dream about colorectal cancer. And so it's easy for me to know the biomarkers for colorectal cancer. If you ask me the current set of biomarkers for, a sarcoma right now, I couldn't tell you. Right. And it is really hard. I think acknowledging how hard it is for oncologists who see, the majority of oncologists see, all tumor types out there, and how hard that really is to stay on top of that. And so I think it's not about. I think it's about just helping to be a partner along with your oncologist to make sure that these areas are being explored and tested. So I think what's the best way to get confidence in the knowledge? I think it's just to ask. Is biomarker testing appropriate, and has it been done, and I think that will kind of trigger a discussion. It'll trigger that biomarker part of the mind that, the visit, there's so many things that have to be addressed in a visit, right? And probably the biggest barrier, if I were to guess about, biomarker testing, a lot of it is just, it's so busy. There's so many things going on. And, sometimes the biomarker just gets neglected. It's not always that, that the oncologist doesn't know, right? It's just that, okay, I need to stop and to think about biomarker testing. So I think that's good. And the second is, I encourage patients to, seek out a second opinion, as well. And that's so critical. It doesn't have to mean that you fly to, New York or Boston or Houston. It can be you go to the oncologist a town over, or, an oncologist in a different group, and then just kind of see if, he or she would agree with the course that you've been on, right? And it's just somebody fresh sitting down to think about your case, to kind of think about what things are there and maybe possible. Those are things that can be really, really helpful. And I think leaves you either with, okay, here's a gap that we can fill with our testing, or, I have confidence now that that all the right testing has been done, right? So I think that's another kind of key component and things that we can, that I encourage for patients as well. And I'll do that for patients that come see me. I'll say, if at any point you feel like you need to, I'll give you a name of, half dozen oncologists that I'd, send my family to for a second opinion as well. And so I think that's always helpful.
Yeah. I'll just say here my own opinion that part of why Dr. Kopetz doesn't even say the words that are clearly coming out of him is that he is a perfect example of this, and that's humility. If somebody thinks they know all the answers, you can be pretty sure that they don't. So I would also just really encourage everyone, do not engage on your first treatment. Certainly if it's a complicated treatment decision, don't engage on that without having at least two opinions, if you possibly can wait. And the reason is, you will just learn so much from having two different conversations. You will hear something that's slightly different, and it'll open up a different avenue of conversation that maybe you go read about. Maybe you have a conversation with another member of One Cancer Place, whatever it might be. I'm a big believer that anytime there is a critical decision to be made in the management of a patient, particularly with metastatic cancer, that they should probably be seeing somebody with some level of expertise in that disease. And Dr. Kopetz didn't say that, so I don't mean to be putting words in his mouth, but I do think, if you're talking about metastatic disease in a particular cancer type, it's really not that heavy of a lift to have a relationship with one expert. It doesn't have to be at the biggest cancer center in the world, just someone who treats only that disease, and go to them for those really critical visits when there's a big decision to be made. But outside of that, you can manage all of your, dosing of your therapy at home. And that model works really well for a lot of cancer centers. So something really to consider. But that doesn't answer the original question, which was about sort of the biomarker. And Dr. Kopetz did talk about that. But Erica asked the question, what about when you say minimal testing, what's sort of an example of minimal testing? And is that a good term or a bad term?
No, and I think it's the I wouldn't use that in a derogatory way at all. Like if you have, for colorectal cancer, right, if you have had testing for your KRAS, NRAS, BRAF, MSI high and, and HER2 status, and maybe an NTRK fusion, but tossed in there as rare as those are. But if you've had that done, that's kind of a minimal biomarker testing that would really align with all the guidelines that are out there. Even if you haven't had 200 genes, you've derived the vast majority of benefit from biomarker testing out there, right? Are there a few scenarios where occasionally we'll find some pol E or some other alteration that could make a difference? Yes, there are. Those are, but the added benefit of going beyond that testing is there. It's just modest. So I think, how do you define that minimal testing? And that minimal testing typically is, what are on the current guidelines, right? What does ASCO recommend? Guidelines for that? What does NCCN guidelines recommend? Now, those are tough. Those are designed for oncology communities. And so you really then have to, rely on, the patient advocacy or, discussion in places like One Cancer Place to say, hey, what are the tests that I should have done for my tumor type, right? To ask some of those questions. Because it's sometimes hard to get access to the ASCO NCCN guidelines necessarily and interpret them. So, but that would be kind of what I would think about is just what's the current guideline standard of care.
Yeah. For anyone who doesn't know where those are, nccn.org is a pretty amazing website. It is not the easiest thing to navigate, but once you find the guidelines, they're a treasure trove. However, they are not the easiest thing even to understand for physicians. So it's because there's an enormous amount of complexity in treatment decisions for cancer, but they're set up to be an algorithm that people can. And Julie just mentioned they do have patient versions. That's true, and so I would encourage people, if you know your cancer type and you know what you're dealing with, it does not hurt to download that PDF and just look through it sometimes and try to figure out where you fit in there and ask some questions. It's a really interesting and helpful resource. We're down to our last minute or two. I wanted to see if anybody has any other just questions want to ask.
This was very, informative, and it aligns with what I'm thinking too, especially the humility part. I mean, if an oncologist can't handle that simple desire for knowledge, then something is wrong. But I think, but then every patient is different as well, and you acknowledge that. Yes, but as a patient, I'm intensely curious, and I want to know more, much more than is needed. So I'm often met with, yes, we can do this, but it will have no consequence at all for your treatment. And as a patient, that's sort of interesting because I really don't care always. I just want to know more.
Yeah. And I'd say there's a whole spectrum, Roger, as you mentioned, of patients. And I think we do have patients like you, and we have that conversation and say, well, I don't know what to do with it, but let's figure it out together. Right. And, and we have a whole, cancer, affects everyone. And there's nothing more motivating to patients, of course, than wanting to try to tackle their own cancer, and understand their cancer better. So I think we do have to be, have to be supportive and encouraging of that type of exploration as well. And so I don't want my comments to say, be careful about the testing. I think if you test and understand that that you're not going to act on it, that's okay. It's, when you do the test and then then somehow you end up doing some treatment that's really a bad idea. That's the harder part, but absolutely. And I think, the amount of information that we can get on these, the tumors that we're dealing with now is well beyond anything it was 5 years ago. And I don't think we as a field have really figured out how to handle all that information yet, but it's a surmountable problem. And I think, as Chris mentioned, in aggregate, this information is going to be incredibly insightful. And I think, just the idea of, how as a community do we learn, how do we enable patients to donate data and tissue and bring it together in some useful way that we can all learn from these, I think is, the next area that we should be exploring as an advocacy community.
Yeah. Maybe a topic for another conversation is using the specific mutation that exists to try to identify what weakness biologically that results in, and then taking advantage of it, whether that's immunologically or with cell signaling or some other therapeutic agent. I think 25 years ago, the idea was, well, we found this mutation. We have to find something that inhibits the signal that comes from that mutation. And while we've had a couple of just enormous, outrageous successes in that, maybe 3 or 4, I guess, the vast majority have been kind of defeating. And what they've really told us is biology isn't that simple. You can't just put your finger in the hole in the dam when there are cracks all over the dam. And I think understanding the biology more completely requires understanding the complexity of the genes that end up being expressed in a tumor tissue that has a particular mutation profile, for instance. And I think that's kind of what's coming. It's what we're kind of alluding to. But the challenge is that we need to be able to identify who is who much, much more predictably. And we need to do that in a large number of patients in order to do that, and then run clinical trials in those patients. And I'll say, the last one of these that we did, we talked a lot about trying to initiate clinical trials in patients with certain gene expression profiles. And those studies, have actually been ongoing for a few years now. But we're still not quite there in terms of either the quantity of patients that are being identified prospectively, the quantity that have been in clinical trials, and the answers to those questions with the right therapies yet. So, and it doesn't mean we're not ever going to get there, but it just takes a few years. For reference, 10 years ago, well, maybe 12, patients were diagnosed with metastatic melanoma, were expected to die in about 9 months, and now that number, on average, is closer to 5 years. It's just an unbelievable delta that can occur, but it is not really incremental, it's exponential. And I believe we've started to hit that exponential part of the curve in terms of impact of these technologies. But the hard part is we're still probably at the bottom of that exponential curve. So we're getting there, but we're not quite, in my opinion, not quite to that inflection point where we're going to see these major shifts just yet, but that can happen at any moment. It just requires more time and patience and doctors being interested. So, Scott, do you want to give the last word, or I don't know if you were planning to stick around for the last 25 minutes, but I assume you probably have other things you need to do.
Yeah, I can certainly, chair. So first of all, just a big thanks. I think just to have a space and an opportunity to have these conversations, to start the conversations, because even an hour for a topic like this generates another 5 hours of follow-up discussions that we have to find time and space to do. But just delighted to do that. And I do want to just leave people, like Chris mentioned, with a sense of optimism. Like, we are making progress, right? And biomarkers, a good biomarker can be just as impactful as a good drug, right? So, and can really make a big difference in how we manage it. But, it's complex, and hopefully this shared some, our perspective on what are the complexities as we feel like we've, kind of gotten the majority of the information we're going to get out of the DNA. And now we're kind of moving down deeper into understanding the tumor, trying to understand the complexities are rising exponentially, but the potential there is also for the rewards to rise exponentially as well, as Chris mentioned. And so we're excited by where the field's going. And, our goal is just to try to get us there as fast as we can.
Thanks, you so much.
I'll just say, I have the privilege of getting to talk to Dr. Kopetz a number of different settings. And every time I have the chance to hear him talk, I learn something new. And I will just say, the thing I said before is so true. You're not going to meet somebody who's more humble. He has made enormous contributions to this space and continues to do so through his lab's work, through his work with patients. And you wouldn't know it from hearing him talk. You'd think he's just one of us normal people doing work all the time, but he really is a true leader. And Scott, I really thank you for doing this. It's awesome. You want to see if anybody else has any questions now that, we're kind of in the more informal part. Anything come to mind while we were talking?
I have a question, but if somebody else does, because I always have questions. So please, if somebody else has a question,
That's
ask.
what we like about you, Julie.
Ask.
Well, I think when you were talking about the interpretation of the biomarkers versus like testing more and more kind of thing, I think that's really a powerful concept. And I guess the question is, in terms of that interpretation, like who and how do we impact that? Because I'll give an example. The other day, a patient got back her testing results, and she's a colorectal patient. At the top, really large, it said negative for HER2, and then it said, X through these are the treatments that have no clinical benefit, and it was a list of 4 drugs, right? And so she gets this back and you're sitting there going, okay, I have this disease. There's only like 4 drugs that might work. And then I have this list of 4 that aren't going to work. So I said, look, 98% of patients that don't have that mutation. So yes, will those not work for you? Yes, but it's not like it's taking things off the table. Those aren't generally on the table. It's like they're not being put on the table, but the way it was reported really was a really disservice, I think. And so if you don't, and if you don't have the time to spend with your oncologist going through all of that, I mean, like obviously the oncologist isn't probably, isn't going to look at it and say the same thing. It's not a big deal, but like, how do we help in terms of that interpretation? Because that just to me is a disservice in terms of the value that the biomarker testing can be and empowering patients with information that can be valuable for when they talk to their clinician. Because, for that patient to waste time talking about, these drugs that don't matter, really, even though it was factual, it just really irked me. And you see that all the time. And it's unfortunate, It
really is. Part of it, and it is getting a little better than it was several years ago, where it was just an unmitigated disaster. And now we're just a small brush fire. It's not a raging inferno, but it's still an area. And I think you highlight exactly, it's kind of the, what is the key message that you need to get across to the patient and the physicians around those biomarkers. And I think there's a few levels, how do you interpret for the standard of care therapies, what are on the guidelines? And then, increasingly, people are and historically have been looking to these panels to kind of think a little outside of the box as well. And it's kind of like that, that next level below the level of guideline approved pairing of biomarker and treatments that are the most complex. And I think where it still is falling apart, right? And so, a few of the issues are that that it's really hard to interpret some of these. KRAS, we've got HER2, we've got ERBB2, we've got those figured out, and we can interpret almost anything that comes on the report. But when you start to get to other genes, it becomes increasingly complex. Not every mutation disrupts the function of the gene, right? So you can get mutations that are don't always have relevance. There are some assays that will sequence lots of genes, but don't sequence the normal, right? So why is that important? Well, kind of getting in the weeds a little bit, but there are, when you see something that doesn't match what an average normal patient's germline sequencing is, well, was that just a variant that you inherited from your patient or from your parents, right? That has nothing to do with cancer. There's no functional relevance. It's just, that's why you've got, some more freckles than someone else, right? Or, all these kind of germline alterations. If you don't sequence the germline, then you don't really know what is different in the tumor, which is potentially relevant, versus what's kind of just something that you inherited and is perfectly fine and benign, right? So that is a really important distinction. And there are still some assays out there, even some big name ones, that don't do the germline sequencing to help figure out what is really different in the tumor or not. And then the second is, when you actually get those, when you find those alterations, does that alteration actually cause functional significance? And that can be, there are thousands, tens of thousands of alterations on, a single gene that are out there. We don't always know what they all do. And so you have to rely on mathematical modeling sometimes, or predictions about whether or not they are potentially doing anything to the function of the gene or not. So that becomes really difficult to kind of interpret. And there, it's a bit of a crapshoot, right? About what's reported in those kind of below the line alterations, and some, some reports now just kind of dump them all in the back, and they just don't even make an attempt, right? They'll just say, here, besides the 5 that are on the guidelines, here are all the others, and you can go do what you want with them. Others will try to make an attempt at that, but it is really hard to stay on top of the literature. We have, we're very fortunate that we have an institute at MD Anderson that I can send a report to, and they've got a army of, I think, 5 PhDs that are scouring the literature full time, right? And updating databases of these alterations, and they can go and they can give an alteration of that. But, that's not a sustainable model and an expandable model. And so a lot of these reports don't really have that depth of information on some of those alterations. So it makes it really difficult to tease out some of those. So I think that, acknowledging what we really know and what we can use, and being very clear, like you said, about how do we best communicate that in a very patient-friendly way, where we're not discouraging or distracting patients. And then, how do you, kind of make sure that we're not providing information that may be misleading by these molecular alterations. I think if I were, king of the world for a day or two, maybe it'd take more than a day or two to fix, but, I think you can need to have a patient-centric report, right? And then a physician-centric report should be in the same thing, right? Page 1 and 2 is the patient ones, and then, 3 and 4 are for the physician, and then the researcher who wants to dive into things, it's pages 5, 6, 7, 8, 9, 10, or whatever, right? That to get all those. And I think that would really help because it's a lot of the things you're describing are how do we break through the clutter for the oncologist to make a big red X, right? That gets their attention. But at the same time, that's maybe not how the message we want to be conveying to the patient, which is just like you described.
These challenges, for anybody who hasn't looked at these reports, I mean, sometimes we'll see these drugs that, in that disease we've known have had no relevance for years. And one wonders if that's like a regulatory component where it's required that the report says a certain number of things just to prove that they validated against certain prediction algorithms or something. But I think what Scott just recommended is actually pretty brilliant. It's kind of what I've always hoped that we would see, is something that's just like straightforward language. Here's what you do with this information. If you're just a general oncologist and you just need to know what to do, here's what to do. If you're an academic and you have these other 8 clinical trials available, and here's some information for people who actually work in the lab and understand some of this stuff at a deeper level, or not just understand it, but actually could take action on it, for instance. So any other questions? I'm sure Julie might have another one, but
I have a comment.
Yeah, go ahead.
What you just were talking about are subtypes in presentation for those reports. So
Yeah,
we're back to the original conversation, which I think is a brilliant idea, Dr. Kopetz. And I wonder if you've ever mentioned it to any of the testing companies.
I have, yes. It's a very complex where there's some, regulations. I think fundamentally the problem is that they get paid for the test and not for the interpretation. All the interpretation is gratis, right? It's kind of like, and I think the business, and because ultimately these are businesses, right? They look at their core business and then they look at this like unreimbursed division over here that's not returning anything. And they're like, well, unless there's enough of an uproar, we're going to put them on a shoestring budget. And, I think if CMS were to reimburse, it all is based on kind of billing codes, right? If they had a billing code for testing and a separate billing code for interpretation, it'd all be solved, and then we would have the, have that. But anyway, it's
not to get political here, but that I have a similar opinion when it comes to certain medical subspecialties as well. Like there are certain parts of medicine that you are reimbursed for doing a procedure. And sometimes what people should be paying more for is the physician who says, don't do that procedure. You don't need it. Because it would save everyone a lot of money and heartache. But and interestingly, that's not too irrelevant when we think about, advanced biologic correlative testing in cancer. Is like if somebody could say, hey, we can do all these things, but we know for certain that it's not going to be useful for you, and we refuse to do it. Or you'd almost be better off sometimes paying somebody to tell you that and being honest about it than being willing to do a bunch of things that are not useful.
That goes along with kind of my other question in terms of like access to specialists is you all like help each other out, and you consult with each other all the time on patient cases, and somebody will ask you a question, whatever, but it's not reimbursed, and it's not, that's just, you just do because it's part of who you are as people. But that to me is like so powerful. And I know Janet has a huge passion too for like rural and people that don't have access to an NCI center. And it's like, how could that become a reimbursable thing? Those kind of consults where it's not a full consult, it's just but you could probably look at 1,000 cases in a day and provide, kind of directionally point people towards the right direction. And you probably already do that. Yeah. But if there was a way to make that part of like specialists kind of, incentive, that they actually got, reimbursed for that, how many patients could be really impacted in a really positive way?
And I think that's one of the disappointing things about COVID was that we had all of this, telehealth that was enabled, right? Across state lines, it wasn't a problem of licensure. Everybody was pitching in together. You could take care of your patients wherever in the U.S. they were, at least. And we have seen 80% of those walls go right back up now. Oh, no. So it's a real shame. Like I can't do a televisit with somebody, across the state line, vast majority of states now, which is a real shame, because that is to Janet's passion and interest, like that was a lot of it, right? We were getting patients that, they weren't going to travel. It was just, a second opinion visit. And that's what that's what was being done. But
who do we lobby to change that, Dr. K?
Yeah, it's all state medical boards. So you got to lobby all 50 state medical boards. So we'll just divide them out today. Yeah. Erica, you want Hawaii? Is that what I heard? Erica, you volunteered down there, but no, it's not my understanding, and I don't know this field well, but I think it's all just medical board decisions we made at state level.
Then the ACS, which parses out each state with ACS CAN, would be the vehicle to address that, I would think.
Potentially, they could help in advocating for those type of things. So,
because we all feel so helpless, there's so much to be done, and where do you start? Who do we work with to influence? And that you just mentioned something hugely significant to everybody. And it's terrifying that this has gone back to having the silos up like that.
Well, and I think too, there's so much effort and attention on multidisciplinary tumor boards, which I think is amazing, and that should continue. But I feel like intradisciplinary tumor boards would be helpful too, because there's community oncologists who just need some input from another oncologist potentially. So, you don't necessarily need the liver specialist and the whatever. You need like within colleagues, if you will. But again, something that wouldn't just be because you guys are nice people doing it for each other, but something that could really be institutionalized to help patients.
Yeah.
Yep. Yeah, Julie, I am aware of a few entities trying to create a business model that would help solve that problem, but it is a big challenge. The only real model I've heard that actually seems like it could make sense is having, high-end care for very, very wealthy individuals, and then using the extra revenue stream to help facilitate that sort of care for more people who need it. But, creating the infrastructure that is required with sort of those initial, sort of like the Tesla model of building roadsters to build slightly less expensive cars to less expensive cars until you finally work your way down, hopefully to more broadly applicable price levels. But it is a real challenge. And I think what's interesting is COVID gave us some evidence, though, that it is possible to break that cycle of these, whether it's state boards or whatever it is, like, in the right setting, we can all do something that actually works better. Why can we not take that information and do something that makes more sense? Obviously, this is a very United States-based conversation because this problem really doesn't exist in Europe. Not nearly the way it does here. And it probably doesn't exist in many other countries quite the same way it does in the United States either, because unfortunately our medical system is designed for profit. And that is the root of the problem. I don't know how else to put it, but that's the root of the problem. So unless we change that, I don't, and there are ways that theoretically we could change it, but people have been trying for about, 50, 60 years now, no luck. So maybe we all just need to take the power of social media and aggregate our voices and get them together. But I did want to get to Janet's question here because it's a really good one. So Janet wrote in the chat, when the data from those less useful biomarker studies are done outside of a clinical trial, is there any mechanism that attempts to collect the information and keep it for when someone might start a study using that biomarker? And so if you anticipate, like, for instance, that everyone might do this RNA-seq test, and that's IO relevant, but to date, we haven't figured out what the right signal is. And then all of a sudden we do figure it out. Can we take that old information for a given patient and pull them back into a clinical trial? Or is there any avenue like that? I think MD Anderson probably has something like this already, Scott, but do you see this happening more broadly in the rest of the country?
Yeah, what a great question, Janet. And that's kind of the key, one of the key things that we hope is in place, and where I do see a bit of optimism. So I think one is we're doing better at getting data in a useful form through electronic health records. Still not perfect. So a long way to go. But where we are now compared to 5 years ago is night and day, right? In terms of kind of getting this data in a useful manner, in kind of discrete elements that we can mine. And I think this idea of kind of big data is going to be great for things like this, right? There are now tools and platforms and efforts that are in place to really help with some of that. So I think 5 years from now, we're going to look back and be surprised that this was a problem, that we couldn't have data in the right place. There'll still be some, not too Pollyannish, but it's something we'll certainly, I think, be in a better position. So what can we do now with that? I think it's, making sure that, pushing interconnectivity of our health records is a big issue, right? This falls into the same category, Chris. We've been trying to do this for 50 years, but we're making progress. The other is, I think, the advocacy, probably where we also have opportunities, is advocating for the biomarker companies themselves. They really care what the advocacy community says, because that there's a component of direct to consumer on all of these the biomarker testing. So I think you guys do have a fairly loud voice with them. And I think asking them to, help open up their treasure trove of data, right? To say, you guys are sitting on 5,000 patients that have had testing done. What are the opportunities to go back and to learn from those, right? From those patients. And I think some of the companies are better than others. There are some that are really making their data available for research studies and are enabling and actually resourcing some of these, right? Which is another level, right? It's not that, we'll share the data with whoever has a good idea, but we're going to help, finance people to, pay for the salaries of the smart people to sit in front of the computer and to help answer these questions then as well. So I think the more that we can push biomarker companies to do some of that digging and to have mechanisms in place, I think that could go a long way as well. So I think the more that we understand from these large data sets, a corollary to what I said before, right? When you go from 5 genes that are binary yes, no to 400 genes that are binary to yes, no to 20,000 genes that are all continuous variables, is that the number of patients you need to get signals starts to go up and up and up exponentially as well, right? So when we start to think about these next generation biomarkers, we're quickly getting beyond the type of studies that academic center X can put together their 100 patients that they've had and report out something useful, right? You need 1,000 patients to interpret this more complex data, if not more. And so that really then pushes it into consortium collaborations, or working kind of with the biomarker company. So I think that's a great place to, Janet, to your point, to really try to push the field some.
Yeah, I just want to help for someone who may not know much or anything about statistics to understand why that's relevant. So that every test that we do, if you assume that there's maybe a 5% chance that it gives you inaccurate information at some level, that means that of every 100 patients, 5 of them will have something that is wrong. It could be wrong telling you to do something or to not do something, right? And in that setting, right, then if you take 10 different questions and you ask 10 different questions, you now have that 5% chance now multiplying up, because you don't know which of those questions you actually are getting incorrect. And that's why when you do multiple questions within the same data set, you actually have the need for more and more and more numbers to eliminate the noise of the sensitivity or specificity problem that test may have. And that's not a super obvious thing to most people, but it ends up being true in the most painful ways. Because if we make a decision on something where we asked 30 questions at the same time and then go after the one thing that came out right, the likelihood that we are incorrect and then follow that path and spend a lot of money and time and resource on it is actually fairly high. And so you really want to limit the likelihood of failing before you start as much as you can. And that's why you need these large numbers of patients in order to sort out the noise when you're asking a lot of complicated questions at the same time. What a fun way to end on a statistics conversation. Everyone loves that. So any last questions before we go?
I have a very brief one because, as a computer scientist and a data scientist, I'm very interested in how we communicate data and use data. So I was just curious if you had any thoughts on how data science and in particular machine learning and artificial intelligence and these developments that we're seeing can be utilized even in research, but also maybe treatment situations where there's too much data for a human to process.
Yeah, and I think these are the areas where we're going to see that next incremental acceleration in big data. I think what we need to get you is good, clean data out of our EMRs to enable this. 70% of our data elements that we would probably want right now are clean. 30% are still really noisy, right? Did the patient have toxicity? How did they tolerate the therapy? Did they switch therapies because they progressed on the cancer, or is it for some other reason, or patient preference? All these things that are written in the note and conversational flow of the oncologist and not as discrete elements. So I think it's about how do we clean the data up so we can get the data to you so that you can make sense of it, and then importantly, how in these how can we have interpretable, actionable modeling that comes out the other side. And I think that's another area where the field of AI is really moving towards, is kind of, an interpretable modeling, meaning that, so that we can start to go back and ask the question, okay, why is that? What was the underlying biology behind that observation that gives us confidence about the finding, and then also, allows us to build on that a little bit more. So I think with better techniques and better data entry, I am incredibly excited by this area.
Yeah, I'll just agree that data scientists have become critical to all of our endeavors. And one of the cool things about incorporating that specialty field into oncology has been that we learn iteratively about all the things we're doing wrong about how we build databases as we go. So if you can visualize in real time where you're making major mistakes in data entry, you can also improve the design of what we call the case report form, the page that talks in the database about a particular question. And it's really interesting and educational for those of us that do this literally every day to say, wow, we really have been doing this wrong for a long time. And it's only because we're incorporating the right people into the field that we get to see that. So that's a plug for data scientists to look into oncology as a place to for jobs, because there's ample need out there. Erica, you want to close us out? Thanks again for a fun conversation.
I just want to make sure that everybody is thanked for having attended. Dr. Kopetz, you are a treasure, and we so appreciate you. And Chris, I want to let you know how much we appreciate you as well. You add so much to every one of these conversations, and we really couldn't do this without you. Thanks, Dr. Scott. Thanks, Dr. Chris, and everybody else who attended today. I'm going to be recording a thank you to our sponsors, but for everybody here, thank you for showing up. This is great. Come to our next one, April 8th.
Thanks, Erica.
Thanks all. Enjoyed it.
Thanks, Scott, so much.
Certainly. All right. Bye-bye.
You're the best. To hear those two minds back and forthing about all the progress that's being made in the search for the cure for cancers, particularly colorectal cancer, which happens to be Dr. Scott's passion. In fact, I tell anybody who asks, if you have a BRAF mutation, get yourself to Houston and see Dr. Scott Kopetz. Thank you so much for joining us today, both Dr. Scott Kopetz and Dr. Chris Heery. Dr. Heery, I want to thank you from the bottom of my heart, and to thank Arcellx, your company, for allowing you or encouraging you to help us in this. I want to tell you a little bit about Arcellx. It's a company that is destroying cancer and advancing humanity. You work for a beautiful company, and I'm sure they're proud to have you, Dr. Chris. So today I'd like to give special thanks to our partners who support us financially so that we can provide these brilliant conversations to the general public. Those strategic partners are organizations, the Colorectal Cancer Alliance and the Colon Cancer Coalition. Both organizations have supported us by emptying their pockets to help us, and we're so happy that they are helping us and believe in us. The Mirati Therapeutics Company is a supportive partner. The Natera Testing Company is another supportive partner for this series of brilliant conversations. And we have been supported from the very, very beginning by our founding partner, the Seagen Corporation. Thank you all so much for joining us today for this really important conversation. Now, before I let you go, please like this YouTube presentation and subscribe to our channel. The sooner we have 100 people that are subscribed, the sooner we'll have our name on this channel. So please accept my invitation to subscribe to the channel. Thank you so much. We'll see you again soon.